ProSite. ProSite is a database of protein families and domains. It consists of biologically significant sites, patterns and profiles which help to map the unknown sequences to known protein families. Since the latest NAR database issue paper, ProSite has increased its number of signatures to 1,559 documentation entries, 1,308 patterns, 863 profiles and 869 pro rules. ProSite was constructed by Amos Bayreuth. It is based on the protein sequences in SwissProt. Certain regions of protein sequences have been conserved in nature, and as a basic thumb rule, these regions form the basis of the function of a protein and or for the maintenance of its three-dimensional structure. The patterns or signatures that these regions of the protein sequences display help in setting them aside from other unrelated sequences. Therefore, it is possible to ascertain certain families of proteins to an unknown sequence based on the signature pattern or profile of the particular domain. The basis of ProSite is regular expressions describing characteristic subsequences of specific protein families or domains. It has now been extended to contain some profiles which can be described as probability patterns for specific protein sequence families. ProSite makes a distinction between patterns and rules which are both described by regular expressions. A pattern is defined as a unique characteristic that belongs to a protein domain family. A rule only displays certain features in a protein sequence that may or may not help in assigning it to a specific protein family. For example, a potential phosphorylation site can be found in many protein sequences. The ProSite databases uses two kinds of signatures or descriptors to identify conserved regions, that is, patterns and generalized profiles, both having their own strengths and weaknesses, defining their area of optimum application. Each ProSite signature is linked to an annotation document where the user can find information on the protein family or domain detected by the signature, such as the origin of its name, taxonomic occurrence, domain architecture, function, 3D structure, main characteristics of the sequence, domain size and literature references. ProSite signatures are also associated with corresponding pro rules which contain information for the automated annotation of domains in the UniProt KB/SwissProt database. Some of the information stored in pro rule can be accessed via Scan ProSite, which provides additional features such as active sites or disulfide bonds associated with a particular domain. The interface of ProSite is as follows. Here is an image which shows the interface of ProSite. The ProSite website was redesigned and new predictive 60 tools were implemented to assign more detailed functional information to the scanned proteins. Users who want to scan their own proteins against all ProSite entries or to scan a ProSite entry against a protein database will find a new version of the Scan ProSite web page. Access to ProSite. Browse ProSite documentation entries. It is possible to search ProSite using the following parameters: post-translational modifications, compositional biased regions, domains, DNA or RNA associated proteins, enzymes, electron transport proteins, other transport proteins, structural proteins, receptors, cytokines and growth factors, hormones and active peptides, toxins, inhibitors, protein secretion and chaperones and others. The entries in the database are alphanumeric and follow a definite structure. Let us now consider an example. PDOC000202 Kringle domain signature and profile. The character in the first column is used to indicate whether a documentation entry is new in this release, plus sign or has been modified to asterisk. The numerical characters in the positions 3 to 7 provide the documentation entry accession number. The numerical characters in the position 9 is used to indicate how many data entries such as patterns, rules and profiles or matrices are described by a documentation entry. Examples are PDO-C 00001 1N glycosylation site, PDO-C 00351 2 disintegrin domain signature and profile, PDO-C 00027 2 homeobox domain signature and profile, PDO-C 000861 thymidylate synthase active site, PDO-C 00160 DNA topoisomerase 2 signature, PDO-C 001092 phospholipase A2 active site signatures. Search by author. Searching ProCite by author name is possible. Search by citation. ProCite can be searched using this option for journals published after 1965 with options such as journal name, volume and year. Search by description. It is possible to search ProCite database of protein families and domains by the description of sites and patterns by using a keyword. The keyword may be any word or partial word appearing in the description of the documentation entries. Search by full text. The text search is the relatively new feature available on ProCite release 19.24. The search is case sensitive. Wildcards may be used to search with partial words. Boolean operators such as and, or and not can be used to restrict search with brackets such as the flower bracket and to specify the order. SRS or sequence retrieval system. ProCite offers two alternative search forms under this category. These are self-explanatory. Tools available at ProCite. Scan ProCite. This tool enables a user to scan a sequence against ProCite or a pattern against SwissProt or TREMBL or PDB for the occurrence of patterns, profiles and rules or motifs stored in the ProCite database. It also visualizes matches on structures with a graphical view and feature detection. Motif scan. It allows a user to scan a sequence against the profile entries in ProCite and PFAM. Motif scanning allows search for all known motifs that occur in a given unknown sequence. InterProScan. This is a Perl stand-alone InterProScan package that allows the user to query the input or unknown sequence against InterPro. The InterPro database integrates ProCite, prints, PFAM, ProDOM, SMART, TIGR FAMs, PIR superfamily, Gene3D and Panther databases. The tool combines different protein signature recognition methods into one resource. PRAT. This is a tool that interactively generates conserved patterns from a series of aligned proteins. The tool may be used to look for patterns that are conserved in a set of sequences. The advantage of this tool is that the user can specify what kind of patterns should be searched for and how many sequences should match a pattern to be reported. PSScan. It is a Perl program to scan ProCite locally. PF tools. These are standalone programs to create and scan ProCite profiles.